Okay, the last part of this lecture that I want to cover is a little bit on the biology of bacteria and just how they're quite different from yeast cells and what we can infer about them. So one of the first things to remember is other than their interior cell structure differences, being that they're prokaryotes, that bacteria are much smaller than yeast, so they're quite a bit harder to see in the microscope. If we look at this image taken of a combination of yeast and bacteria in a microscope image, the yeast cells are here and they're fairly visible and large, but the bacteria cells are quite small and they're basically these little lines. We would call them we call rod cells or bacillus that are seen here on the image, and so that gives you an idea of the difference in size between a bacterium and a yeast cell. So the bacteria themselves are classified in one large way by differences in their cell wall. So we would subdivide bacteria by what were called gram-positive and gram-negative cells. And so you may hear those referred to in different contexts. And it's a measure, or it's determined by staining the cells, and they are stained with a process called the gram stain. It involves two different stains that are applied with an alcohol or ethanol washing step in between. And the main measure between gram-positive and gram-negative cells, the distinction, is that on a gram stain, if it's done correctly, the gram-positive cells are purple because of retention of the first stain that is used in that process called crystal violet. The gram-negative cells are, in a gram stain slide, are pink, and that's because they have been washed free of the first stain, crystal violet, and then they're stained by the second stain used called safranin. And so the main biological difference between these is that gram-positive cells have a very thick layer of the cell wall substance of peptidoglycan. And gram-negative cells have a thin peptidoglycan cell wall, but they have also an outer membrane layer. The types of cells that are usually of concern in brewing as contaminants or spoiling the beer are primarily gram-positive cells. The gram-negative cells don't do well at the low pH and in the anaerobic environment or in the presence of the alcohol and resins from the hop. So it's primarily gram-positive cells that spoil beer. However, you can have some problems from gram-negative cells on occasion. How do bacteria reproduce? Kind of similar in a way to yeast, in that they reproduce asexually. It's a different name, though. We call it as a process of binary fission. And typically when a bacterial cell divides, it's not like it's a bud that forms, but the cell divides equally. They have just a single chromosome, and so they have to copy that chromosome so that they have two of the single chromosome. And then by the cell separating in a process called cytokinesis, it makes a division between the two areas of where the chromosomes are, and eventually you have two identical cells. So if you're growing bacteria, and if they're growing a lot and you're in a liquid medium like a broth, the bacteria will reproduce enough, and they're in such a great number that they'll turn the medium cloudy. And so this is typically how one would look at a test to see if you just have a liquid medium, if it's supporting the growth of bacterium, if it turns cloudy like that, if it becomes turbid. If you're using an agar plate in a petri dish, then bacterial cells will typically form a colony. And so this picture shows little colonies of lactobacillus. Yeast cells will also form colonies on agar plates as well. And when you have a colony, it's typically assumed that if it's a distinct isolated colony, like just one of these, that originally it came from just one single bacterial cell that began to divide asexually. And so the assumption then is that from a colony, you have millions of just genetically identical cells. And this is useful because then you can sometimes pick a single colony of a bacteria, or if you happen to be working with the yeast, the same thing. You can pick a single colony and isolate it and propagate it, and that would give you an isolated strain. So the concerns of various bacteria in brewing, the ones of most concern are those that are the lactic acid bacteria. And these will produce lactic acid as a fermentation product. So one of the main ones is the bacteria that are species of the genus or the group Lactobacillus. This is the most common bacteria that can be found to spoil beer, and attributes that allow it to do that is because it's resistant to the resins that come from hops that have a bacterial cycle or that kill other bacteria, but Lactobacillus can tolerate those hop resins. Also that they can survive and proliferate in an anaerobic environment that fermenting wort or beer becomes. And because they're fairly easily found, so they can be found on people, they can be found on grain, both of which have close contact with wort that could allow it to be introduced. So Lactobacillus, if we can look at in an image here, are rod-shaped cells. They're gram-positive, and when they produce lactic acid, this can give tartness to the beer, and so it may give it a taste similar to sour milk, because many times sour milk is because Lactobacillus have grown. They could also contribute higher levels of diacetyl to the beer. And yet at the same time, Lactobacillus may also be introduced or used purposely to create sour beers. And this is a growing trend, or has been growing. And so there are many brewers out there that will make use of Lactobacillus to create sour beers. Another organism is called Pediococcus. This is also a lactic acid bacterium. But it has a bit of a different shape. It has a spherical shape, and it's often more frequently seen in lagers, and it may exhibit a late stage growth in the fermentation of a lager. So Lactobacillus and Pediococcus are two of the primary bacteria that are concerned in brewing as spoilage. We can also have a problem from acetic acid bacteria. These come from, they're not directly related to the lactic acid bacteria. They're from a different genus, many from the genus Acetobacter. And these are a problem if you have oxygen in your finished beer. So what can happen is that the acetic acid bacteria can perform a secondary fermentation. They can ferment ethanol, so they use ethanol as basically a food source. And by fermenting it, they produce acetic acid. Acetic acid is what is vinegar. And so you can have, if you have too much oxygen in your beer and you happen to have acetic acid bacteria in it, they can proliferate and turn the beer sour with a vinegar taste. And then lastly, we can have some bacteria of the genus Zymonomonas. Zymonomonas are also able to ferment sugars. They happen to produce acetaldehyde as well as hydrogen sulfide. And as we'll also discuss in the fermentation unit, hydrogen sulfide can be produced by yeast as well. So lastly, just some parting thoughts on bacteria. Bacteria themselves are everywhere. They are ubiquitous, and so it's important to be conscious of contact with any kinds of surfaces in the brew house that might introduce bacteria to your wort or your beer. Any kinds of objects, tap water itself, and even the air can harbor bacteria that are drifting around. And so if one is trying to grow bacteria selectively to test your wort or your beer, you need to make sure, because you may use non-sterile water to make that bacterial test agar or media, that it needs to be sterilized. It needs to be sterilized in an autoclave or a pressure cooker. Only these two devices are able to get the temperature high enough to kill the bacteria and the bacterial spores. Just boiling alone, unless you do it in a very particular way where you go through several rounds of boiling, you cannot usually get adequate sterility to be sure before you or when you produce your agars. When looking for the production of, or the presence, of bacteria, and you're wanting to use a selective agar, you would typically use a test media that contains cyclohexamide. Cyclohexamide is a toxin that it prohibits the growth of brewer's yeast. and it allows bacteria like Lactococcus and Pediococcus and wild yeast actually to grow on your media. So this cyclohexamide is found in some of the different microbiological test augers that you might use in the lab. One of those is HLP. This is called Sue's Lactococcus Pediococcus media, and it's used to test for the presence of those two organisms. When used, it is done in a semi-solid agar tube, and so you add the beer to it, and because it is in a tube that's fairly deep, it will provide anaerobic conditions in the tube, and so that's how you can select for Lactococcus and Pediococcus, which are anaerobes. If you're interested in testing for gram-negative bacteria that can ferment lactose, you would use an agar called McConkey agar. This can detect bacteria such as E. coli that would be those gram-negatives, and you don't want those in your beer. They can be bad. E. coli, in some conditions or in some strains of it, can even be pathogenic, and so one wants to often ensure that the beer doesn't have those gram-negative bacteria in them. If you just want a general purpose agar to determine the number of any bacteria that can grow in beer or wort or even on your yeast stocks, you may use universal beer agar. And so this is non-selective for just looking at any numbers of bacteria that are there. So I would refer you to look at the yeast book, pages 232 to 240 for more information. So thank you for viewing this unit. This is the last slide, the end of it. I know it was long. Hopefully you took a break somewhere in the middle of it. And I will, along with this module on our learning platform, I will include some guidance on some of the important parts as a study guide for you to focus on which parts are going to be covered on the end of unit exam for this. So with that, I will leave you and will rejoin you for the lecture on metabolism of wort by yeast. Thank you very much.